After studying this module, you shall be able to know the importance of inorganic elements, categorize the inorganic elements according to their roles in the biological system, learn the function of important elements such as iron, calcium and copper, identify the general aspects of storage and transport of metal ions, know the role of metals in medicine. Bioinorganic chemistry constitutes the discipline at the interface of the more classical areas of inorganic chemistry and biology. Although biology is generally related to organic chemistry, inorganic elements are also important to life processes. Table lists the essential inorganic elements together with some of their known roles in biology. Bioinorganic chemists study these inorganic species according to their function in vivo. Inorganic elements have also been artificially introduced into the biological systems as probes of structure and function. Heavy metals such as mercury and platinum are used by X-ray crystallographers and electron microscopists to help elucidate the structure of macromolecules. Paramagnetic metal ions have been valuable in magnetic resonance applications. Metal containing compounds have been used not only as biological probes but also as diagnostic and therapeutic pharmaceuticals. The mechanism of action of platinum anti-cancer drugs, gold anti-arthritic agents and technetium radiopharmaceuticals are some currently active topics of investigation in bioinorganic chemistry. Bioinorganic chemistry thus has two major components: the study of naturally occurring inorganic elements in biology and the introduction of metals into the biological systems as probes and drugs. Peripheral but essential aspects of the discipline include investigation of inorganic elements in nutrition, of the toxicity of inorganic species, including the ways in which such toxicities are overcome both by natural systems and by human intervention, and storage and transport of metal ions in biology. Metal functions in metalloproteins. Metals are commonly found as natural constituents of proteins. Nature has learned to use the special properties of metal ions to perform a wide variety of specific functions associated with life processes. Metalloproteins that perform a catalytic function are called metalloenzymes. Metalloproteins are a class of biologically important macromolecules and account for nearly half of all proteins in biology. They are responsible for performing some of the most difficult yet important functions, including photosynthesis, respiration, water oxidation, oxygen transport, electron transfer, oxygenation and nitrogen fixation. Function of metalloenzyme. Metalloenzymes are a subclass of metalloproteins that perform specific catalytic functions. A net chemical transformation occurs in a molecule termed as substrate being acted upon by the metalloenzyme. Some remarkable transformations for which no simple analogues exist in small molecule chemistry under comparable conditions include the catalytic reduction of N2 to NH3, that is nitrogen fixation, the oxidation of water to dioxygen and the reduction of geminal diols to monoalcohols, that is reduction of ribonucleotides. Classification of metalloenzymes is done as per their function. Within each category, there are usually several kinds of metal centers that can catalyze the required chemical transformation, a situation analogous to that already encountered for respiratory proteins. The reasons for this diversity are shrouded in evolutionary history, but most likely include bioavailability of a given element in the geosphere-biosphere interface during the initial development of a metalloenzyme, as well as pressure to evolve multiple biochemical pathways to secure the viability of critical cellular functions. Communication roles for metals in biology. Metal ions are used in biology as triggers for specific cellular functions and to regulate gene expression. Studies of these cellular communication roles are an exciting frontier area in bioinorganic chemistry. Magnetotactic bacteria use magnetite Fe3O4 as internal compass for navigation of microorganisms. They orient on the Earth's magnetic pole and when transported to opposite hemisphere become disoriented and swim upward. Some bees, homing pigeons and even humans are also believed to use magnetite in their brains for orientation purposes. Alkali and alkaline earth ions, especially sodium, potassium and calcium, are used in biology to trigger cellular responses. The firing of neurons by the rapid influx of sodium ions across the cell membrane and the regulation of intracellular functions of calcium binding proteins such as calmodulin are two examples of the phenomena. In fact, calcium has been referred to as a second messenger since primary signals such as binding of hormones to the cell surface are converted into changes in the intracellular concentration of this ion. Among the most recently discovered metal ion classes in biology are the zinc fingers that occur in many proteins which regulate transcription. Interactions of metal ions and nucleic acids. Metal ions also interact directly with DNA and RNA. Some of these interactions are rather non-specific. For instance, the stabilization of nucleic acid structures by sodium and magnesium ions through electrostatic interactions that shield the charged phosphate groups from one another. Recently, more specific bindings of metal ions to the nucleic acid has been discovered. Thus, magnesium and other divalent metal ions serve as cofactors for activating catalytic RNA molecules, and monovalent cations such as potassium stabilize the structure of telomeres units that terminate the DNA double helix at the end of chromosomes. The relative stability of these structures may be dictated by the concentrations of sodium and potassium in the cell. Finally, some inorganic based drugs such as cisplatin act by coordinating directly to DNA and metal complexes, have been used as cleaving agents to probe the tertiary structures of nucleic acids. Metal ion transport and storage. How do metal ions get into cells and how are they stored? This topic is an active area of investigation in bioinorganic chemistry. Although it cannot be classified with others according to metal function, the most thoroughly studied metal in this respect is iron. Iron enters bacterial cells following chelation by low molecular weight compounds called siderophores that are excreted by the bacteria. In mammals, iron is bound and transported by serum protein transferrin and it is stored by pterin in most life forms. The nearly spherical hollow shell of this latter protein has the capacity to bind up to 4,500 ferric ions. Details about how iron is passed among these proteins are incomplete and under active investigation. Copper is transported by the serum protein ceruloplasmin, and another such protein, albumin, is also known to bind the transport metal ions. Metallothionein is a cysteine-rich protein that is expressed in large amounts when excess quantities of certain metal ions, including toxic ones such as cadmium or lead, are present in cells. Metallothionein thus serves as protective role and may also be involved in control of metal transport, storage and concentration under more normal conditions. General aspect of storage and transport of metal ions. Charged ions must pass through hydrophobic membrane. Neutral gases such as oxygen and carbon dioxide and low charge density ions such as anions can move directly through the membrane. High charge density cations require help. Once inside the cell, metal ions must be transported to the location of their use, then released or stored for later. Release from ligand is often not trivial. Storage requires additional molecules. Iron storage and transport. Iron is essential in small amounts for both plant and animal life. However, it is toxic in large quantities. Biologically, iron is the most important transition element and it is involved in several different processes. As an oxygen carrier in the blood of mammals, birds and fish, for oxygen storage in muscle tissue, as an electron carrier in plants, animals and bacteria, cytochromes, and for electron transfer in plants and bacteria, ferredoxins. For storage and transport of iron in animals, as nitrogenase, as a number of other enzymes, aldehyde oxidase, catalase and peroxidase and succinic dehydrogenase. Iron enters bacterial cell following chelation by low molecular weight components called siderophores that are excreted by bacteria in most life forms. In mammals, iron is bound and transported by serum protein transferrin and it is stored by ferritin. Iron storage. Iron which has been released into the cell must either be used immediately for biosynthesis or stored in a safe form. In humans, iron is stored mainly in the bone marrow, spleen and liver. About 10% of all the iron in the body is in storage. Two proteins are involved in iron storage, ferritin and hemosiderin. Ferritin is a huge hollow spherical protein with a wall mostly made up of alpha helical peptide chains. Fe2+, ferrous, is oxidized and transported into the ferritin interior and deposited as an iron mineral core, traditionally described as ferrihydrite, which is attached to the inner wall of the sphere. Up to 4,500 iron can be stored, although the normal value is about 2,000. Hemosiderin. Hemosiderin is another storage form for iron in organism, in particular during iron overloading. Hemosiderin was first isolated from horse spleen in 1929. Although the structures of iron cores of ferritin and hemosiderin are similar, the protein component of hemosiderin is largely unknown. The iron-protein ratio in hemosiderin is even higher than in ferritin and is assumed that this insoluble species is formed via lysosomal decomposition of ferritin. Protease in the lysosome degrade the protein shell of ferritin. The released iron core dissociates and reforms as the amorphous hemosiderin. Transport of iron, transferrin. Transferrin are iron-binding blood plasma glycoproteins that control the level of free iron in biological fluids. Transferrin binds iron very tightly but reversibly. Contains two specific high-affinity iron 3 binding sites. The affinity of transferrin for iron 3 is extremely high but decreases progressively with decreasing pH below neutrality. Transferrin without iron, known as apotransferrin. Once carbonate binds in this site, the metal binding site is fully organized so that iron 3 can now coordinate in an octahedral, oxygen-rich environment of ligands favorable for such a relative hard metal ion. Calcium. Function, signal pathways, example muscle contraction, skeletal material. Concentration, outside the cell, the concentration of calcium ion is 0.001 molar. Inside the cell, it is less. Calcium 2+, ATPase in cell membrane controls the concentration. Calcium storage. Calcium carbonate in a protein matrix makes up eggshells and coral skeletons. Calcium hydroxyapatite in a collagen framework is the stored form of calcium in bones and teeth. Collagen is triple helix fibrous protein. Hydroxyapatite crystallizes around the collagen. When needed, calcium can be released and reabsorbed. Calcium pump. Calcium pump are ATPases that transport ions using energy obtained from hydrolysis of ATP across membranes. Calcium ATPases are members of P-type family of ion pumps which are responsible for the ATP-dependent active transport of ions across a wide variety of cellular membranes. The protein is far less stable when these calcium ions are removed. It was solved by adding a drug molecule that binds near the calcium binding site and freezes the protein into a stable but non-functioning form. The calcium pump is an amazing machine with several moving parts. It is found in membrane. It has a big domain poking out on the outside of the sacroplasmic reticulum and a region that is embedded in the membrane, forming a tunnel to the other side. When an ATP breaks, calcium pump transfers 2 calcium ions through the membrane and 2 or 3 hydrogen ions back in opposite direction. In this process of pumping, a phosphate is transferred from ATP to a special aspartate amino acid in the pump number 351. The switching is controlled by large motions of the ATP binding domains which push and pull on the proteins surrounding the tunnel, opening and closing it appropriately. Permanent activation of the pump by calmodulin is physiologically as harmful to cells as its absence. The concept is now emerging that the global control of cell calcium may not be the main function of the pump. In some cell types, it could even be irrelevant. The main pump role would be the regulation of calcium in cell microdomains in which the pump co-segregates with partners that modulate the calcium message and transduce it into important cell functions. Copper function: oxygen transport, oxygen activation, electron transfer, availability, third most abundant transition metals, iron in the organisms, 300 mg in human body. Storage of copper: after its hepatic uptake, copper may be stored with hepatocytes, secreted into plasma or excreted in bile. The biliary route represents the major excretory pathway of copper and largely accounts for its hepatic turnover. Copper retained by hepatocytes is mostly bound to specific metal binding proteins, primarily metallothionein, or incorporated in several cuproenzymes. Impairments of homeostatic mechanisms in brain copper mechanisms have been associated with neurodegeneration in human disorders such as Menkes disease, Wilson's disease and Alzheimer's disease. Copper transport: intake of copper at doses that exceed physiologic demands are normally met with efficient homeostatic mechanisms. Ceruloplasmin, albumin and transcuprin, and to a lesser extent certain amino acids, are major copper transporting constituents in circulating plasma. Studies with radioactive copper have established that after intestinal absorption, copper appears in portal bloodstream bound to albumin and to a much smaller extent amino acids. This copper is rapidly transported to the liver. Considering the number of copper atoms massed within its structure, ceruloplasmin can be considered to be an effective vehicle for shuttling copper out of the liver and delivering clusters of copper atoms into a variety of non-hepatic cells and tissues. Metals in medicine: many first encounter the use of metals in medicine as well as metal toxicity through literature. The use of iron and copper can be traced to the ancient Greeks and Hebrews through their writings. Lewis Carroll's Mad Hatter suffered from mercury poisoning. Among metal ions used over the centuries were Hg2+ for the treatment of syphilis, magnesium for intestinal disorders and iron for anemia. These early examples represent crude approaches, the refinement of which did not until recently begin to match in sophistication or efficacy the contribution of organic chemists who introduced sulfur drugs, penicillin and mechanism-based inhibitors such as methotrexate. One of the leading anti-cancer drug is cisplatin. Cisplatin administered by intravenous injection for the treatment of testicular, ovarian and head and neck tumors. Cisplatin cures testicular cancer when diagnosed early enough in more than 90% of the cases. Aurinofin, where TTAG is tetra-O-acetyl-thioglucose, is a first orally administered drug for the treatment of rheumatoid arthritis. It is an important member of the class of anti-arthritic gold agents, the other of which are injected. The third example is technetium, the metal in which technetium is supplied as its 99M technetium radioisotope. This class of complexes is selectively taken up by myocardial tissue and has proved to be excellent for imaging the heart. Platinum, gold and technetium, three non-essential transition elements, have found a place in medicine. Given the range and the variety of inorganic compounds, the potential application of inorganic chemistry to improving human health are boundless. This aspect of bioinorganic chemistry, now in its infancy, seems destined to be an area of rapid and significant growth. Anti-cancer therapeutics. Cisplatin was the first metal-based medicinal agent to enter into worldwide clinical use for the treatment of cancer. Figure displays various platinum-based anti-cancer agents. The platinum-2 of the cisplatin unit binds covalently to deoxyribonucleic acids, DNA, more specifically to the N7 of either guanine or adenine in the dinucleotide sequences GG and AG to form interstrand crosslinks and 1,2 or 1,3 interstrand crosslinks. Figure represents some of the antimicrobial and antiparasitic metallodrugs. Summary: bioinorganic chemistry includes the study of both natural phenomena such as behavior of metalloproteins as well as artificially introduced metals, including those that are non-essential in medicine and toxicology. Metal ions are used in biology as triggers for specific cellular functions and to regulate gene expression. In mammals, iron is bound and transported by serum protein transferrin and it is stored by ferritin. Hemosiderin is another storage form for iron in organism, in particularly during iron overloading. Calcium pumps are ATPases that transport ions across the membranes using energy obtained from the hydrolysis of ATP. Major copper transporting constituents in circulating plasma are celluloplasmin, albumin and transcuprin, and to a lesser extent certain amino acids. Metal coordination compounds in therapy has opened an array of possibilities which traditional organic or biological molecules cannot fulfill any longer due to growing drug resistance. Metallodrugs hold still tremendous potential to help mankind over drug resistance and to find new cures in medicine.